Oh, good morning, everyone. And my name is Sibing Wen. I'm honored to be the moderator of this session, along with my colleagues, Professor Junyi and yeah, Yixuan and Fu Yang. And I'm honored to introduce the first speaker, Professor Nippon Chadibakong from Chennai University, Thailand. Professor Nippon is a highly accomplished MD PhD investigator and a physician scientist who really had a numerous contribution to the field for both basic translational and clinical research. And today he's going to deliver a talk on the mitochondria related contribution to chemotherapy-induced cardiotoxicity. Professor Nippon, welcome.
Okay, thank you so much, Yibin. Do you hear the slide well? Hey, you see the slide well?
Yes, yes.
Okay,
Thank you.
good. So, first of all, I would like to thank the organizing committee for having me here, sharing with you some of the knowledge that we have been studying regarding chemotherapy-induced cardiotoxicity. As all of us may already know that cancer has been the burden in most countries around the world. And it has been reported that in 2040, there'll be around almost 30 million new cases of cancers being detected. And for that numbers, around 15 million mortalities could be encountered. For the therapeutic management of cancer, we know that there is like chemotherapy and also radiotherapy. These two are conventional cancer therapies. And at this time, we also have gene therapy and targeted therapies, which is an advanced and innovative cancer therapies. Nevertheless, doxorubicin is still the conventional chemotherapy that has been shown to be high efficacy in treating cancers. Doxorubicin is the FDA-approved anthracycline, and it inhibits topoisomerase 2, which promotes DNA damage and cancer cell death. And it has been used to treat the malignancies and solid tumors for many decades. Despite its high efficacy, it has been known that doxorubicin can also create cardiotoxicities, which can lead to the irreversible cardiomyopathy and also heart failure. Studies have shown that doxorubicin could cause a severe production of the reactive oxygen species, leading to the excessive oxidative stress in our body. It also shown that it can cause mitochondrial injury in the cardiomyocytes, such as increased mitochondrial RS production, mitochondrial dysfunction, including mitochondrial depolarization and also mitochondrial swelling. And more importantly, it also causes mitochondrial dynamic imbalance. All of this could lead to the cardiomyocyte death and leading to cardiac dysfunction, finally leading to the heart failure. I would like to talk a little bit about the doxorubicin-induced mitochondrial dynamics imbalance. Studies, including ours, have shown that after receiving doxorubicin in the heart, the level of mitochondrial fission was significantly increased, whereas the level of mitochondrial fusion in the heart was significantly reduced. This already caused the mitochondrial dynamic imbalance in doxorubicin-induced cardiotoxicity. Interestingly, in the past few years, we asked ourselves, what if we could inhibit mitochondrial fission to prevent its increase in the heart and also to promote the mitochondrial fusion in the heart after receiving doxorubicin? Could this prevent doxorubicin-induced cardiotoxicity? To test this hypothesis, in this experimental protocol, we use male Vista rats And these rats receive doxorubicin according to this timeline. And also the interventional groups, we provide the MDV1, which is the mitochondrial fission inhibitor, co-treated with doxorubicin. And also another group we provide with the M1, which is the mitochondrial fusion promoter, co-treated with doxorubicin. And then at the end, we determine we're using the echocardiography and also heart rate variability. And then after that, we euthanize the rats and study the oxidative stress, cardiac biomarkers, mitochondrial function and dynamics, and also cell death. Now let's move to the results of this study. First, I would like to talk about the LV function in these DIC rats. This slide shows the ejection fraction and also E/A ratio. You see that rats receiving doxorubicin, the ejection fraction was significantly reduced, and also the E/A ratio was also reduced too. Interestingly, rats being treated with either MDV1 or M1, you see that this reduction in the ejection fraction and also E/A ratio was successfully reduced. When we look at the HRV or heart rate variability, we measure the similar to the Holter monitoring, and then we determine the low frequency and high frequency. Low frequency normally indicates the sympathetic and parasympathetic activity in the heart, and also high frequency indicates the parasympathetic activity. In this kind of investigation, we determine the LF per HF ratio. And if this ratio is increased, it indicates the cardiac sympathovagal imbalance. Let's look at the results. In this study, doxorubicin caused the increase in LF per HF ratio, indicating that doxorubicin caused a severe cardiac sympathovagal imbalance. Interestingly, doxorubicin co-treated with either MDV1 or M1, you see from the graph that the level of the ratio was successfully reduced compared to the doxorubicin alone. All of these findings suggested that inhibiting fission or promoting fusion could successfully improve cardiac function and autonomic balance in DIC rats. How about the oxidative stress? We measured the serum MDA and also tissue MDA in the heart of the rats being treated with doxorubicin. As you can see here, doxorubicin creates a significant increase in the MDA level in both the serum and also tissue, cardiac tissue. And rats being treated with either MDV1 or M1, you see that the level of MDA in both the serum and also the cardiac tissue was successfully reduced. We also look at the cardiac injury biomarkers. In this case, we look at the NT-proBNP and also troponin 1. Not surprisingly, doxorubicin also caused the increase in the NT-proBNP and troponin I in this case, and MDV1 or M1 could successfully reduce these adverse biomarkers in the heart. These results suggest that inhibiting fission or promoting fusion reduce oxidative stress and also cardiac injury in DIC rats. Let's move to the mitochondrial dynamics in these DIC rats. In this study, we determined the level of DRP1, which is the marker of the mitochondrial fission, and also MFN2, which is the marker of the mitochondrial fusion. As you can see here, that doxorubicin caused an increase in DRP1, indicating an increase in mitochondrial fission, and also reduced MFN2, indicating the reduction of the mitochondrial fusion. In this case, MDV1 or M1 could successfully reduce the fission and also promote the mitochondrial fusion in the heart of these DIC rats. How about the cardiac apoptosis? We measured the Cliff Class-B3. In this case, we found that doxorubicin caused an increase in the Cliff Class-B3, and MDV1 and M1 also successfully reduced these biomarkers. And we also look at the tunnel positive cells. In this case, as you can see, doxorubicin caused an increase in the number of tunnel positive cells, and doxorubicin co-treated with either MDV1 or M1 successfully reduced the number of these tunnel positive cells. All of these findings suggest that inhibiting fission or promoting fusion prevented mitochondrial dynamic alterations and cardiac apoptosis in DIC rats. So for this section, in summary, doxorubicin induced cardiotoxicity by increasing the level of lipid peroxidation and also cause and mitochondrial injury as indicated by the mitochondrial dysfunction, mitochondrial dynamic imbalance, and by increase the mitochondrial fission DRP1 and reduce mitochondrial fusion biomarkers MFN2. This could lead to increased cellular apoptosis in cardiomyocytes, leading to cardiomyocyte death and resulting in cardiac dysfunction and leading to heart failure in the end. By treating with either MDV1 or M1, which is the mitochondrial fission inhibitor or mitochondrial fusion promoter, it could successfully reverse all of these adverse markers and resulting in the improved cardiac function in DIC rats. That's about doxorubicin. Interestingly, in case of breast cancer, normally, if the HER2 positive is found, then in addition to doxorubicin, targeted therapy is always treated with the trastuzumab. Trastuzumab is the targeted therapy, which is the humanized monoclonal antibody. It has been used to treat the early or metastatic HER2 positive breast cancer, overexpressed HER2 breast cancer type. It has been shown sporadically that trastuzumab itself can also cause cardiotoxicity. However, this has not been strongly confirmed in the previous study. Therefore, we look at the effect of the trastuzumab on the heart in the trastuzumab-induced cardiotoxicity model. And it has been shown that trastuzumab caused an increase in lipid peroxidation similar to the doxorubicin and also caused mitochondrial injury, increased oxidative stress, mitochondrial dysfunction, and also, more importantly, it also caused mitochondrial dynamic imbalance. All of this could lead to cardiac apoptosis, cardiomyocyte death, and also cardiac dysfunction and heart failure. So, since trastuzumab can also cause an increase in mitochondrial fission and reduce mitochondrial fusion, leading to mitochondrial dynamic imbalance, we ask ourselves if by promoting mitochondrial fusion using M1 or inhibiting mitochondrial fission by using MDV1, could this protect trastuzumab-induced cardiotoxicity? In the experimental protocol, we also use these rats and giving trastuzumab according to this timeline, and also co-treated with either MDV1 or M1. And then we determine the echocardiography and heart rate reliability, euthanize and study these biomarkers. First, let's move to the LV function in these TIC rats. Similar to doxorubicin, as you can see, the ejection fraction and also the EPA ratio was successfully reduced using the trastuzumab. Interestingly, by treated with either MDV1 or M1, you see that ejection fraction and EPA ratio was successfully restored. We look at the LF per HF ratio as well, and as you can see, trastuzumab caused an increase, markedly increase in the LF per HF ratio, suggesting that it caused the severe cardiac sympatovagal imbalance. And MDV1 and M1 successfully reduced this ratio. This result suggesting that inhibiting fission or promoting fusion could successfully improve cardiac function in the TIC rats. Then we look at the cardiac stress and injury in TIC rats. As you can see here, cardiac MDA was markedly increased in the trastuzumab in treated rats, and also level of troponin I and NT-proBNP. By treating with either MDV1 and M1, it successfully reduced these cardiac injury biomarkers. This result suggests that inhibiting fission or promoting fusion also reduces oxidative stress and cardiac injury in TIC rats. For the mitochondrial function, we look at the mitochondrial RS production, mitochondrial depolarization, and mitochondrial swelling. As you can see here, that trastuzumab itself caused a severe production of mitochondrial RS production and also caused the reduced red per green ratio indicating mitochondrial depolarization and also reduce the absorbance indicating mitochondrial swelling. Interestingly, treating with either MDV1 or M1 could successfully restore all of these mitochondrial function markers. For the mitochondrial dynamics, we look at the mitochondrial fission marker DRP1 and also mitochondrial fusion marker OPA1. Similar to doxorubicin, trastuzumab caused an increase in DRP1, indicating the increased fission, and also reduced OPA1, indicating reduced mitochondrial fusion in the heart. MDV1 or M1 successfully restore these mitochondrial dynamic markers. These findings suggest that inhibiting fission or promoting fusion restore mitochondrial function and dynamic regulation in TIC rats. Last but not least, we look at the cardiac apoptosis in TIC rats by looking at Bax level, cytochrome C, and also ClIP-Cas3. You see that trastuzumab increased Bax, cytochrome C, and ClIP-Cas3 level, and MDV1 or M1 successfully reduced it. For the tunnel positive cell, trastuzumab, as you can see here, it also produced an increase in tunnel positive cell, and treating with either MDV1 or M1 reduced these positive cells. These results suggest that inhibiting fission or promoting fusion attenuate cardiac apoptosis in TIC rats. In summary for this section then, trastuzumab caused an increase in lipid peroxidation as indicated by increased level of MDA, and also caused mitochondrial injury as indicated by mitochondrial dysfunction, mitochondrial dynamic imbalance. And all of this could lead to cellular apoptosis, cardiac myocyte death, and also cardiac dysfunction and heart failure. By treating with the MDV1 and M1, all of these adverse parameters were successfully reduced, leading to an improved cardiac function in TIC rats. So in conclusion, inhibiting mitochondrial fission and promoting fusion effectively protected the heart from doxorubicin and trastuzumab induced cardiotoxicity by attenuating oxidative stress, mitochondrial dysfunction, mitochondrial dynamic imbalance, and apoptosis, leading to improved cardiac function. The modulation of mitochondrial dynamics could be a novel therapeutic target for preventing doxorubicin and trastuzumab-induced cardiotoxicity in the future. I would like to thank all of these granting agencies for supporting these studies. And last but not least, I would like to thank all the CERT members who dedicate their time and effort in making these studies possible. And for that, thank you so much for your kind attention.
Thank you, Professor Limpong, for this very clear presentation. It's very interesting. Any questions from the panel? Maybe I can start. Just a quick question. I understand that many of the, particularly doxorubicin, the cardiac oncology drugs actually can reduce cardiac mass in terms of the weight of the heart and actually the weight of the whole body. And so I wonder, when you observe the protective effects from the heart failure, have you also observed the kind of a restoration of the weight and the prevention of the loss of the mass?
Thank you, Yibit. That's a good question. Yes, we also look at the weight at the end, and we found that compared to the doxorubicin treatment alone, the treatment with MDV1 or M1, at the end, the weight of the animal, the reduction of the weight is less than the reduction of the weight in the doxorubicin treated alone. Yes. And for the heart, of course, because it can prevent or attenuate the level of cardiac apoptosis and cell death, so the heart is also not, that bad compared to the doxorubicin treatment. Mm-hmm. Yes.
Okay. Well, okay. Any other questions from the panel?
Hello, Professor. So maybe I can ask.
Yes, please.
Hello. So thank you for your talk. It's really very wonderful. So I would like, since I don't have much knowledge on the heart, so can I ask, do you see any effects? So in addition to on the cardiomyocyte, on other cardiac cells, does the drug has any effect on the other cardiac cells?
I see. I think you want to know about the endothelial cells, right?
Yeah, and fibroblasts and stuff. Yeah.
Right. I have been asked by similar question so many times. Yes, we plan to, but in this study we haven't looked at all of those yet. Yes.
Okay, thank you.
But I'm sure that particularly the endothelial cells, it has been damaged a lot, and I still wonder, but I still believe that the intervention with either mitochondrial dynamic alteration like this could at least attenuate the damage to the endothelial cells.
Probably the cardiomyocyte is the major effect is still on the cardiomyocyte on the phenotype.
Yes.
Professor Yishen, I think you have a question.
Yes, Dr. Nippon, it's very nice to hear about your talk. I'm Yishen from Peking University. So I have a very quick question that what do you think can be used clinically to modulate mitochondrial fusion in the future?
I see.
Yeah, that's my question. Thank you.
Yes. We also ask ourselves about this question too. There are several current conventional medicine that can also affect the mitochondrial dynamics similar to M1 and MDB1. However, the potency of those conventional medicine is not as strong as these two drugs. Therefore, the level of protection is not that much, unless, I don't know, if we have to use a very high dosage in order to compare with the efficacy of the either M1 or MDB1 that we use here. However, to develop MDB1 and M1 into the clinical use, I think it's going to take a lot of time because, of course, it has some side effects as well. You will know that.
Yeah, I'm curious if there's kind of an antibody drug conjugates that can help to enrich more of this drug in the heart.
Right, that will be interesting. Yes.
Yeah, I think that's probably going to be the key, is to limit those,
toxicity.
actually remove, hopefully to target the cancer drug only for the cancer, and then protect the heart only in the cardiomyocyte. That would be the best.
Right. That's the ideal one.
Yeah. All right. Thank you.
Thank you.
Yeah, we can move on to the next one. Thank you.
Okay. So it is my honor to introduce the second speaker, Professor Xiaoyu Tian. Please show the introduction of Professor Tian. So Professor Tian is an assistant professor of the School of Biomedical Science at the Chinese University of Hong Kong, and she received her PhD from the CUHK Department of Physiology. And after graduation, she continued as a postdoc in the Cardiovascular Research Institute in UCSF, and she later joined the CUHK again as a research assistant professor in 2015, and now he is assistant professor. And her current research interests include the regulation of vascular cell function by circadian genes, endothelial cell regulation during vascular inflammation and atherosclerosis. And her research work has been primarily funded by the RGC of Hong Kong, and she received funding from the National Natural Science Foundation of China, the category B of the Young Scientist Fund in 2019. As a corresponding author, she has published numerous research papers in journals such as Circulatory Research, Cardiovascular Research, PNAS, and Advanced Science. And she has also co-authored over 100 papers. So let's welcome Professor Tian to deliver her talk today. Thank you very much.
Okay, thank you, Junyi. So now I'm sharing my slides. Desktop. So just let me, so you can see my slides, right?
Yes.
So just let me know if the network is unstable, because I get a pop-up message showing that. Okay, so I would like to thank Junyi and the organizing committee for inviting me to participate in this conference. So today I'm going to talk about the nuclear receptor PPAR in endothelial cell function. So to this audience, so I don't think we need much introduction on what is endothelial cell dysfunction. So in the past, we have been focusing quite a lot on these factors, so vasodilators and vasoconstrictors that are released by the endothelial cells and causing the vascular tone-related endothelial dysfunction. So you get impaired vasodilation and enhanced vasoconstriction, and then this could be one contributor to many major cardiovascular disease. And also, in addition to these, we also have some inflammatory and thrombosis-related factors that are also produced by endothelial cells and causing endothelial dysfunction as well. And as we all know that endothelial dysfunction can occur in major organs, so not only heart, but also like kidney, eye, and also other distal organs. And this is the result of the contribution from many cardiovascular risk factors, so those common things, and also now we know that aging and some other uncanonical cardiovascular risk factors also contribute to the endothelial dysfunction and leading to major disease. So our work started by looking at the regulation of vascular tone vasodilation and vasoconstriction. And what we found is that in the diabetic and obese mice induced by the diet, we can see an impaired flow-induced vasodilation in the small resistance arteries. So as we can see here, so if you have a vasoconstriction by the alpha-1 agonist, and then you see a reduced diameter of the resistance arteries. And then if you give flow, then you have a dilation. So we call this flow-mediated vasodilation. And in the obese and diabetic mice, you don't get much dilation. But then if we give the PPAR-D agonist GW compound, then we see a almost restored flow-mediated vasodilation. So by the compound, so indicating that this may be a beneficial compound. And so later we had a CDH5 Cre-induced deletion of PPAR-D in the endothelial cells. And we used the ischemic model in the hind limb, so in the legs. And over time, you will be able to observe the post-ischemic vascular recovery. So you see coming back of the flow, so by the color. And in the knockout, so we see a much reduced restoration of the perfusion. And so this is the image. And in addition to that, we also see less angiogenesis along the side. And in addition, so we can also see that the functional restoration of vasodilation indicated by the acetylcholine-induced vasodilation in the femoral arteries is also being restored as well. And if you look at the vasculature in the muscles, what we can see is that the restoration of the endothelial cells, the capillary cells by the endothelial cell marker V Heering is impaired. And at the same time, we also see the tight junction marker 01 also get less. And this contributes to the impaired barrier function. So as we can see, the leakage of the Evans blue color. So it's much darker in the knockout mice. And because of this impaired vascular barrier, so what we see is that a lot more of the vascular inflammation, so indicated by the infiltrated macrophages in the muscles as well. And mechanistically, we focus mostly on the hypoxia-induced factor. So because this is one major factor being functional for the vascular repair. So after these ischemic injuries. So what we see is that if you have PPLD, you have a less degradation of the HIF-1 alpha. So as we show here, so if you inhibit the PPLD, then you get less HIF-1 alpha expression. And if you have more PPLD, then you can stabilize the HIF-1 alpha protein. And the molecular study also showed that, so this interaction of PPLD on the HIF-1 alpha is mostly on the ligand binding domain side of the PPLD. So, so that also explains why in some of our data set, if we give a ligand, it doesn't really improve the vascular recovery too much. So because probably this is one reason. And on one side, so the ligand binding domain of PPLD interacts with the HIF-1 alpha. And on the other side, so on the HIF-1 alpha side, what we see is that we have two here. So we have the ODD and TDD. So we can also see that the ODD domain is participating in this effect. So if we mutant that part, then we get much less of the interaction. So this indicates a direct interaction of PPLD on HIF-1 alpha that helps to stabilize HIF-1 alpha after its expression in the endothelial cells in this kind of post-ischemic injury. And so later we also used an ICOM2 promoter-mediated expression of the PPLD in the endothelial cells using the AAV method. So what we have here is that if we re-express PPLD in the endothelial cells locally in the muscles, so we can see a recovery of the perfusion and also an enhancement of the perfusion in the wild type. And if we do that in the if we look at the vasculature in these muscles, we can also see an enhancement of the VK herring, so indicating that the vasculature has been restored and improved. And also there's a reduction of the inflammatory markers VCAM1 as well, as we can see by the treatment. And in the obese and diabetic mice, what we see is very similar. So if we have an re-expression of PPLD in the fat mice, so what we see is an enhancement of the proliferating endothelial cells, so indicating an angiogenesis. And along this, we can also see a lot of these angiogenic related genes are upregulated by the treatment. So all these as we know that they are related to the angiogenesis function of the endothelial cells. And meanwhile, we can see a reduction of those inflammatory genes as well. So indicating that not only we have more endothelial cells, but these cells are functional and they don't have much inflammation for after the post-ischemic injury for repairing the vasculature. And when we did this work, so we did some of the, we did a single cell sequencing of these endothelial cells. And what we see is that we have those classical genes that indicate the angiogenesis function has been impaired in the knockout, and more inflammatory genes that are upregulated. And along the side, we also see some genes that are related to endothelial junction and adhesion function that are being significantly affected. So especially in the capillary endothelial cells in the muscles. And because this post-ischemic model get a mixture of a phenotype, so you get angiogenesis, you get inflammation, and also you get these junction-related genes being affected. So we want to have another model to see whether these barrier-related function can be functionally important in other disease contexts. And so here we chose the sepsis model, or in the mice, we can use the experimental acute lung injury model to study this function. So that is because when we have this barrier disrupted in the endothelial cells, you get not only the proteins coming over to the interstitium, but also you get all these inflammatory cells, these neutrophils and monocytes to get into the interstitium and causing more inflammatory response. So we used a classical acute lung injury model. So using LPS, what we see is that in the knockout, you get much less survival. And at the same time, you get a lot more of these inflammatory cells, the inflammatory monocytes increase in the lungs that doesn't go away. And at the same time, you also have a persistent increase of these inflammatory cytokines, so IL-12, IL-6, and along with others. And if we use both either the lipopolysaccharide-induced injury model, or if we use a polymicrobial sepsis model to see whether the effect is similar. So what we see is that effect is quite similar. So you get a persistent inflammation that doesn't resolve over time. And also, if we use the inducible Cre comparing to the constant Cre, we get a very similar response of these vascular leakage indicators, the vicedestrin, so it doesn't improve over time. And if we look at the pulmonary endothelial cells using markers, so this one indicates it's the capillary endothelial cells, the GPI protein. And what we see is that we get much less the tight junction protein claudin 5 in either all the endothelial cells or in the capillary endothelial cells, and also at a protein level as well. So indicating that this could be one of the downstream junction proteins that mediate this response. And if we use the single cell sequencing again to look at all the cells within the lung after injury and in the knockout versus wild type. So what we can see is that the interferon related signaling are very high in the knockout animals, so after the injury. So if we look at individual genes, then what we can see is that the JAK-STAT signaling, interferon, and all these are being highly upregulated. So this could be one of the genes that mediates the downstream phenotype and identify from these general capillary endothelial cells. And if we take a further look at these capillary endothelial cells, so what we see is that a group of the cells that are particularly induced by the interferon, so these this subcluster here. And out of all these cells, what we see is that a lot of the chemokines, so including CXCL10 and 9, and all these are being highly upregulated. And if we look at whether this chemokine is expressed on the capillary endothelial cells, what we can see is that it is expressed by endothelial cells. So they have a lot of this chemokine. So showing that it doesn't really only coming from immune cells, but the endothelial cell themselves can produce a lot of these chemokines. And mechanistically, what we found is that in the lung microvascular endothelial cells, if you have less PPLD, you get an overactivation of the STAT1. So this is one major transcription factor that would lead to overproduction of the CXCL10. And if we directly treat the cells with CXCL10, we see a very discontinued endothelial barrier, so the tight junction barrier and the adherent junction barrier in the cells. And this is because the CXCL10 can directly cause the ubiquitination of these junction proteins. And as showed by here, so we see more ubiquitination. And if we use a protein degradation inhibitor, we get more expression of these tight junction proteins. And On the other side, so this chemokine can also mediate the T cell chemotaxis. So in the in vitro assays, to here as we showed that if we inhibit CXCL10 itself by the neutralizing antibody, or if we use the chemicals to inhibit its receptor CXCL3, you can see reduction of the chemotaxis as well as the transmigration function of the T cells across these endothelial cells. And in vivo, so if we use either the antagonist against the receptor, or if we use the neutralizing antibody, we can see that even after the symptom developed after LPS injection for a few hours, so if we do a treatment, we can still see an improvement of the lung function. So you get less edema and you get more airspace. And also at the same time, you get less of the inflammatory cells infiltrating in the lungs, and also an improved barrier function of the endothelial cells, along with the upregulation of those tight junction markers. And so this is the summary of this part. So I think, so am I a little bit over time? So because I can't see on my screen. So in fact, I still have a few slides to go if it is okay. So in the first part, we have showed that the PPLD is important for mediating the endothelial barrier function in the microvascular endothelial cells. So we also want to know whether the macrovascular endothelial cell barrier function is being affected by PPLD. And for this part, we used a disturbed flow enhanced atherosclerosis model by doing a partial ligation of the left carotid artery here, and this would enhance the disturbed flow. And also what we see is a unstable plaque formation, quite a lot, very severe in the left carotid arteries. And with this, so we analyzed a few data set, and what we see is a stress granule related protein that are very high in the not only in the mouse cells but also in the human cells. And from our own model, what we see is that we get a amplification of these RNP granule-related pathways, so especially this G3BP1 protein here in the endothelial cells. And if we look at the endothelial cells in the mouse aorta that are exposed to either laminar flow or the disturbed flow, what we can see is an upregulation of this G3BP1 in the areas that are exposed to disturbed flow. At the same time, we also see an upregulation of the PPLD as well. And also they kind of form a biocondensate kind of morphology. And in fact, so if we expose the endothelial cells to the disturbed flow, we can also see some morphology that looks like biocondensate formation. And if we overexpress this, so if we have this PPLD knockout, then what we see is that if we enhance the disturbed flow, we see a lot more of the plaque formation. And also in these areas, we see a damaged vascular barrier so that we can see more leakage of the microsphere beads that are across these endothelial monolayers. And also, if we upregulate the G3BP1, we can see more plaque formation. And also we can see a lot more of this plaque formation, and along with the high expression of G3BP1 in the plaque endothelial cells. And with the help of my friend, Professor Zhou Jin, we detected the biocondensate formation of PPLD and G3BP1. And what we can see is that under the disturbed flow, they will form biocondensate. And also, if we do a bleach assay, you can see its recovery. And this indicates that the PPLD protein may be associated with the G3BP1 granules and forming biocondensate in the endothelial cells where they are exposed to this disturbed flow. And the mechanistically, we found that the ligand binding domain of PPLD is most responsible for this interaction. And so in the last slide, I would like to thank all the collaborators, maybe on this work and maybe as a part of the previous work, and the lab members who did most of the work, and along with some others, and the funding support. And questions are welcome. So thank you, Jenny.
Thank you very much. Yeah, thank you very much for Professor Tian for the wonderful talk about the PPARD and the functions of endothelial cells. So is there any questions that we can discuss? Fuyang, open your audio.
Fuyang, we
didn't
cannot
open
hear
your,
you.
Yeah, you didn't open your audio.
Sorry.
Okay.
Can you hear me? Is it
Yes.
okay? Professor Tian, a very good talk, and it's very interesting to see the interaction between the PPAR delta and the HIF-1 alpha. I just wonder, will the PPAR delta will affect the HIF-1 alpha transcriptional regulation function as a nuclear receptor in the genome, or will it just affect the HIF alpha protein stability? Thank you.
Thank you, Fuyang. That's a question that, in fact, I think we did some mRNA expression and we didn't see any change. So that is why. So because we see first is the protein level change. So then we detected mRNA, and then we did not see much change. So that's why we try to look at the degradation. And that's I think that's your question, right? So transcription level change.
You know, the PPAR delta is a nuclear receptor, and it can regulate some transcription factor function. I just wonder whether the PPAR delta can interact the HIF-1 alpha in the genome and regulate the HIF-1 alpha transcriptional function.
So at least in the endothelial cells, the translational level of HIF-1 alpha doesn't really change, but protein level do change a lot. But I don't know, maybe in other contexts, in other cell types, if you have some other epigenetic mechanism involved, maybe it will change. Yeah.
Okay, thank you. Thank you very much.
Professor Tian,
Yes.
thanks for sharing with us your interesting talk. I am interested in the translation of this kind of findings, particularly in the obese mice that you studied. You know that we have studied the PPAR gamma agonist in the past because it has been used in our patients with insulin resistance and also diabetes. And although it's so effective in treating diabetic, but later on we and others found that it also has a serious side effect on the heart. So my first question is that in obese mice of yours, have you looked at the metabolic alterations? Did PPAR delta agonist that you use change the metabolic profiles in the diabetic mice or obese mice?
Okay, thank you for the question. So in fact, so I think one reason is our treatment is usually not very long term. So usually it's not very long. So that's why we don't see any metabolic profile like lipid and cholesterol and glucose getting very observable changes after the treatment in our mice.
I see.
I think
How
one
long?
reason is
How long did you treat? If I may ask.
For this experiment, usually we do just 1 or 2 weeks. We don't do very long treatment. So for the AAV, because it is injected locally in the muscle, so we did not do a systemic.
Oh, I see. I see.
And then most of the experiments are done within 1 month, so that's why we don't see much change at a systemic level.
Okay, so
But
so
I
it's just the
believe what
intra.
your concern is actually very valid. So there are reported side effects of this compound, right? And systemic level, especially on the heart. So yeah, so.
Okay, all right, thank you.
Yeah, okay, because of the limit of time, we have to move on. Thank you again, Professor Tian, and we can discuss later. Yeah. So let me transfer the moderator to Professor Guo.
All right. It's my honor to introduce Dr. Yibing Wang. He's currently the professor in diabetes and director of a Singapore research program at Duke NUS Medical School in Singapore. He has made a lot of achievements about discovering new regulators of cardiometabolic diseases, and more recently on amino acid signaling and nutrient signaling. Let's just deliver the talk. Let's just welcome Yibing to give us this talk about metabolic targets for HFpEF.
Thank you, Professor Guo, and also thanks the Asian Heart Society for giving me this great opportunity. And today I would like to share with you some of the update on the metabolic targets for HFpEF therapy, particularly targeting on the amino acid. And so here's just my disclosure, and let me see. As we know, the heart disease is actually not getting better, but getting worse. This is a slide I borrowed actually from Joe Hill, where the metabolic burden increase actually correlate with the recent uprising of the heart cardiovascular disease, both burden and disease cause of death. And in particular, the rising of the heart failure actually it's probably more related to heart failure with preserved ejection fraction, the so-called HFpEF, and it's because of the current standard therapy for HFrEF do not have any significant efficacy against the HFpEF. This disease is now recognized as the most significant unmet needs in modern cardiology. And so just to make it very quick, that we have made a lot of progress, as a field to understand the mechanism of HFpEF, and particularly involving 3 key cellular features. One is a metabolic defects, and the other one is inflammation, and the last one is pathological hypertrophy. So the question is whether any one of them can be targeted as a therapeutic intervention for the disease. And so today I will have time only to talk about the metabolic disorder, but there are many other aspects of the feature that we are actually targeting right now. Earlier, we published a work on the glucagon receptor antagonist against the heart failure with preserved ejection fractioning, and this was actually interesting in the sense that this particular therapy is now in phase 2 clinical trial for diabetes. So this is another similar approach as the SGLT2, which actually showed very significant clinical efficacy as well. So this shows that the power of glucose targeted, glycemic targeted control, potential impact on HFpEF. And recently, we performed metabolomic profiling of the HFpEF patient cohorts, and this is in collaboration with University of Northwestern in Sanji's group. And from the serum samples, we identified a number of metabolic modules that are associated with the very severe outcome of HFpEF patients. And in this case, the yellow module, when we look at the composition in the yellow module, and evidently we see glucose, and as well as several other metabolites, particularly the series of amino acids enlisted here, the valine, isoleucine, and isoleucine and the leucine are these three amino acids belong to the same family of branch amino acid. And the similar effects we actually already observed in the HFrEF and also HFpEF by David Heslop in Johns Hopkins. And so what are the BCAA? They are the essential amino acids, and they are the three amigos of valine, isoleucine, leucine, share the similar catabolic pathways, and also some of the shared downstream and unique and shared downstream nutrient signaling effects. And recent studies from Chris Newgar and our lab actually, and several others actually now contribute to the knowledge that a BCAA defect is actually a common underlying mechanism for a number of diseases, chronic disease as well as cancer. In this case, we actually utilize the Joe Hill model, identify that the BCAA catabolic mechanism are impaired. You can see the increased accumulation of a branch amino acid as well as the ketone acid in the serum as well as in the tissue associated with the change of molecular activity indicating the phosphorylation, which is indicative of its catabolic flux of this pathway. And so we think that this indicates that we can utilize this defect maybe as a potential therapy. So here we're using the the pharmacological knockout with BD2, which inhibit the BCK kinase activity and increase the BCAA and BCK catabolism, reduce the BCAA and the BCK accumulation in the tissue as well as in the serum. And you can see that at functional level, we achieved a very remarkable reverse of diastolic dysfunction. Remember, this therapy was actually implemented after 8 weeks of combination treatment, meaning that this is in an established model that we demonstrate a therapeutic outcome. And from the mechanism perspective, we're still, the whole field actually is still trying to understand, but nevertheless, we observed the oxidative stress induced by the ketone acid, as well as reversal of the oxidative stress induced protein acetylation. So we think that this is probably related to underlying mechanism of protein homeostasis. So this result actually led to the clinical trial of BCAA target for HFpEF and HFrEF. And here is the Pfizer's compound, and they actually developed this compound that targeted to the BCK. This is in phase 2 trial right now. And the Brano is actually a company that we just spin off this from Duke and US this year. And we actually have, believe, the best-in-class BTK inhibitor for HFpEF as well. And so I'm going to just skip this. This is actually should be done. So the BCAA target, we believe, will contribute to the current available therapies for SGLT, such as SGLT2 inhibitor and the glucagon receptor, to target the metabolic dysfunction for HFpEF. And we know that the HFpEF is a part of the aging disease. And in fact, BCAA defect is also a major contributor to healthy longevity and the lifespan. And so we, in our clinical as well as the preclinical samples, we see that aging actually contribute to the defect in catabolic activities of BCAA, accumulating of BCAA, and so forth. And if you, we actually see that the BCAA target could be a very important potential a target for therapy for aging associated heart failure as well. And here, just to show that if we impair the BCAA, the heart will spontaneously become prematurely impaired in systolic function. And also in the eyes, we see the features of retinal epithelial cells defects, almost like they are premature aging. And so this is almost equivalent to 15 months like aged white type animals. And so, this is just looking at the morphology as well. So we believe that BCA probably contribute to the heart path, but also can be targeted as a potential therapy. And hopefully, as I said, we'll have chance to actually discuss about our additional progress made in the inflammation and hypertrophy regulation for the disease. With that, I want to thank our collaborators, especially the eye disease with Professor Xiaomeng Wang from SERI and our funders. Thank you.
It's a wonderful talk. Wonder if there are questions.
Hi, Eben. I have just one question. Very interesting regarding the alteration of the branched-chain amino acid in the HepPef that you presented. I just wonder, have you looked at the gut microbiome and see if the alteration in the gut microbiome is also associated with the branched-chain amino acid change in
so
those
there was
HepPef?
a paper, indeed, Nippon, that's a great question. And there was a paper published quite a few years ago showing that the BCAA metabolic defect is associated with diabetes and insulin resistance. And part of that contributing factor is actually from the gut biota. So indeed, the particular strain of gut biota actually can promote the BCAA conversion or production from our gut, because they are essential amino acid. We can only take BCAA either from food or by produced from the gut biota. So this could be another potential mechanism. Yes.
I see. So have you even tried to alter the gut microbiome and see if the
we
efficacy is better?
Yeah, we haven't done that yet.
Okay, very interesting. Thank you.
Thanks.
So I have a question for Professor Wang. So there's a lot of studies now focusing on the field of aging, and it's a complicated status. Do you think there is a driver that commonly induce the downstream effects of aging that can, such as the BCAA metabolism dysfunction?
Yeah, I mean, if I know that there's a common one, then I'll be living forever. And so I think they probably very complex, and metabolic disorder or this dysfunction may be part a very significant one. And so we do see the BCAA is a very conserved lifespan regulator that being demonstrated from fly, from C. elegans, all the way to monkeys. And so I think that this is probably a very strong one.
Yeah. Yesterday I hear a talk from Dr. Ruiping Xiao. She is now making an antibody that can achieve that function, I think. Yeah, but hopefully that she can get that into the industry. Yeah.
That's wonderful.
May I ask a question?
Yes, please. Yeah.
Professor Wang, very impressive, and we have started BCAA follow your step, and I just wonder whether the dietary restriction of the BCAA is a feasible strategy to treat HepF or aging. Are there anyone try to doing this?
Yeah, thanks. So BCAA is actually one of the most common ingredient in food, and of course, particularly in a protein diet such as meat and so it's actually very, very, very challenging to restrict, because then you limited the other protein facts that you can potentially actually cause muscle loss, and so it's very challenging to use dietary control of a certain amino acids. And I think this therapy has a potential to reduce the BCA loading. And we now know more that it's not a BCA, it's actually the building of the BCK, perhaps contributing more towards the pathogenic outcome. So it's we think it's still the intrinsic catabolic activity or restoring this metabolism is probably a better long-term strategy. Thanks.
Thank you so much.
All right, we thank again for Dr. Wang's beautiful talk. So now let's hand the invite the moderator Fuyang Zhang to invite the last speaker for today's session.
Okay, it's my great honor to introduce the next speaker, Professor Feng Zhang from the Sun Yat-sen University. He is a very famous expert in vascular and lymphatic biology. Today he will talk about endocellular adherence and vascular diseases. Welcome. Professor Zhang, you can share your slides. Okay, thank you very much. Professor Zhang, can you hear me? You can start your speech.
Sorry, excuse me，不好意思，张教授，您能这个调换一下，就是在显示设置那里，因为您是两个屏幕，现在这个是演讲者模式的屏幕，您上面那个显示设置，在您左上角有一个显示设置，显示设置下拉，对，显示设置下拉，对，切换，是的，OK，可以了，谢谢。然后张教授，您这边应该是没有说话还是没有声音？请问您可以听到吗？
Can you hear me now?
Yes.
Yes, now we can hear you well. Thank
Sorry
you.
about that. Sorry about that. All right. So is that working now?
Okay, Professor Zhang, you can start your speech.
Sure. Okay. Sorry about that. So first of all, I would like to thank Junyi for inviting me to participate in this meeting, and I also want to thank Fuyang for hosting my talk. So today I would like to talk about endothelial cell adhesion and vascular remodeling. So as we all know, endothelial cells form the inner wall of blood vessels, and the individual endothelial cells require some physical support from cell matrix adhesion and cell-cell adhesion to form the uniform and continuous vessel wall. For example, integrin-mediated focal adhesions can anchor endothelial cells to the basement membrane of the blood vessels to provide external structural support. And the cell-cell junctions can seal the gaps between endothelial cells and prevent free leakage of fluid and macromolecules from out of the blood vessel. So this is basically a fundamental cellular function that maintains tissue homeostasis, including endothelial cell homeostasis. So due to its importance, if cell adhesion is impaired, many vascular diseases will occur, including aortic dissection, hemorrhage, atherosclerosis, diabetic retinopathy, and etc. So today I would like to talk about two stories that we developed recently. The first one is about pulmonary hypertension, and the second one is about retinal macrovascular lumen formation and blood perfusion. So we end up with a common magnetic insights I mean, we found impaired cell adhesion can explain why, I mean, the first and the second, I mean, in the first story, which is related to SMAD4 deficiency, and in the second story, which is related to GMC deficiency. So we end up with a common mechanistic insight related to cell adhesion. That's why I put this project together to talk about this. So for the first story, which is related to pulmonary hypertension, so we all know pulmonary hypertension is a severe cardiovascular disease. There is no medication, currently there is no medication to prevent or to reverse the development of pulmonary hypertension. So it remains a severe disorder. Clinically, there are 5 groups of pulmonary hypertension, group 1 through 5, and they have common pathological features. I mean, when pulmonary hypertension occurs, the distal vessels in the lung will be covered by actual smooth muscle cells, and the inner layer of the blood vessel will get thickening. So this will increase the resistance of lung blood vessels and increase the burden of right heart. That is a key physiological feature of this disease. So for the group 1 pulmonary hypertension, previous studies have located several genetic mutations on BMPR2 receptor, BMPR2. We know, BMPR2 is a component of TGF beta signaling pathway, and there are many other co-receptors, including ALK1 and anaglin, and the downstream signaling components, including SMAD1, SMAD9, and mutations have been found in all these kind of signaling components, and there are, I mean, they are related to the development of group 1 pulmonary hypertension or pulmonary arterial hypertension. However, SMAD4 is also a key downstream effector of pulmonary, is also a key downstream effector of BMPR2 signaling or TGF beta signaling. So, but the role of SMAD4 in group 1, or I mean, in general, the pulmonary hypertension phenotype is not known. So we aim to address this question. So before we started that, we actually did some literature searching, and we found some study already demonstrated that if SMAD4 is deleted in vascular smooth muscle cells, the mice will develop aortic aneurysm and dissection. While during development, if SMAD4 is deleted in the endothelial cell compartment, the mice will develop arteriovenous malformation or AV shunt. But the precise role of SMAD4 in vascular remodeling during pH progression remains unclear. To address this problem, we created a global inducible SMAD4 knockout mice. Why we did that? Because SMAD4, I mean, if SMAD4 is deleted embryonically, it's lethal. I mean, the mice cannot be born. So that's why we created this inducible global knockout mice. And we injected tamoxifen between weeks 3 and 4. You can see during following tamoxifen induction, the mutant mice developed a lot of phenotypes. The mice, the mutant mice were very small, and they don't have fur coverage. And they also developed a pH phenotype, including increased RVSP, increased right ventricular hypertrophy and increased muscularization of distal pulmonary vessels. These are all typical phenotypes of PH. So from this set of experiment, we actually answered the question, SMAD4 deficiency leads to a spontaneous PH phenotype. So then we want to ask what is the cellular mechanism behind. To answer that, we created conditional knockout, I mean cellular specific conditional SMAD4 knockouts. So the first one is smooth muscle cell specific SMAD4 deleted mice. We used MYH11 ERT2 to mediate what's called a smooth muscle cell specific deletion. In this mice, we did not find the PH phenotype, but rather severe aortic dissection and aneurysm phenotype. So this is actually consistent with previous studies using another smooth muscle cell specific Cre SM22 Cre mice, but the mice did not develop PH phenotype. That means we can exclude the possibility that smooth muscle cell specific SMAD4 deletion develop PH. So we excluded this possibility. So then we moved to endothelial cell specific smooth muscle deleted mice. So in this case, we used CDH5 ERT2 to induce endothelial cell specific knockout. So again, we inject tamoxifen in these mice between week 3 and 4. I mean, overall, the mice looks like the mutant mice looked like similar to the control mice, but still there was also significant lethality. About 1/3 of the mice died following tamoxifen induction in 3 weeks. So in this mice, we did not find a severe phenotype with the aorta. There was only a small dilation in the aorta diameter, but importantly, we found the EC specific SMAD4 knockout mice have PH phenotype. So that means the EC specific SMAD4 deficiency is responsible for the PH phenotype observed in the global knockout mice. So then the next question is, what is the molecular mechanism? To answer this question, we did a simple experiment. We did a single cell RNA-seq. So in pulmonary vascular endothelial cell population, we located a class of cell adhesion related pathways. I mean, both cell matrix adhesion and cell adhesion were altered upon SMAD4 deletion. So then we set up a class of in vitro and in vivo experiment to validate this bioinformatic data. So first, we tested whether SMAD4 deficiency affect endothelial cell junction and vascular permeability, and the answer is yes. In the mutant mice, no matter normoxia condition or hypoxia condition, we found severe. We found significant vascular leakage out of the blood vessel in the lung of the mutant mice. Electron microscopy and immunofluorescence also confirmed that endothelial cell SMAD4 deficiency led to disrupted the ultrastructure of annotated cell junction. So this defect lead to macrophage infiltration and inflammation. So we think this is one of the mechanism underlying the pH phenotype observed in the SMAD4 knockout mice. In addition to cell-cell adhesion, we also found SMAD4 deficiency indeed also impaired annotated cell matrix adhesion. For example, in electron microscope, we found some annotated cells are detached from the underneath from the base memory underneath the annotated. So this data suggests indeed SMAD4 deficiency impairs annotated cell matrix adhesion, and we also found extracellular matrix synthesis is also impaired upon SMAD4 knockout or knockdown in vitro or in vivo. So basically, this set of data confirmed that annotated cell specific knockout mice is annotated specific SMAD4 knockout is responsible for the pH phenotype observed in SMAD4 global knockout mice, and the cellular mechanism involves impaired annotated cell adhesion and cell matrix adhesion. So for the molecular mechanism, we actually did a lot of study, but I just briefly talked about the results. So basically, we found integrin beta 1 decreased integrin beta 1 may be responsible for that phenotype. So we found the SMAD4 knockout or knockdown inhibits integrin beta 1 activation and signaling in annotated cells, and we created this EC specific integrin beta 1 knockout mice to we tried to phenocopy that, and we found the annotated integrin beta 1 deletion also led to a spontaneous pH phenotype, and the pH phenotype is much more severe under hypoxia exposure. So this phenotype is similar to what we observed in SMAD4 deleted mice. So to rescue the phenotype caused by annotated SMAD4 deletion, we employed MMP inhibitor, because MMP inhibitor can actually reverse annotated matrix adhesion defects. It can also increase cell adhesiveness. So and our results suggest MMP inhibition indeed can attenuate the pH phenotype in annotated SMAD4 knockout mice. So this is the take-home message that annotated cell, but smooth muscle cells SMAD4 deficiency promotes pulmonary hypertension, and this was due to this disrupted annotated cell adhesion and cell matrix adhesion, right? So I don't have much time left, so I will go through my second project quickly. So for this project, we focused on another molecule called junctional adhesion molecule C, GMC. GMC has been, it has been reported that GMC can promote angiogenesis while its activity in maintaining cell-cell junction. It can also promote immune cell annotated migration, and also there are some other functions related to GMC. So in our study, we try to address whether GMC can promote retinal macrovascular maturation and blood perfusion. So in this case, we also set up the global knockout mice for GMC. In this case, we created the GMC deletion embryonically. So and we found GMC deletion indeed inhibit retinal angiogenesis to some extent. So the effect is relatively mild when the mice were young or were very small. However, if the mice grew up, by P14, we found the phenotype was much more severe in GMC knockout mice because retinal blood vessels get mature from P10. So by P14, we found this severe phenotype. That means GMC might be involved in the regulation of retinal macrovascular maturation. We also confirmed this result using another inducible knockout mice. Again, this is a global inducible mice, and we found similar result. So this is all related to the morphology of blood vessels. How about vascular function or blood perfusion? If whether GMC deficiency also affect blood vessel perfusion? To answer this, we set up an experiment. So basically, we just injected isolectin before conjugated with Alexa 647 in the mice. And if the endothelial cells are perfused by the blood vessel, then endothelial cells can be labeled by this dye. Following this, we dissected the retina and stained the tissue with another color of isolectin before. So if the two channels can overlap properly, that means blood vessels can be perfused by blood very well. If the two channels cannot be overlapping very well, that means blood perfusion is defective. So the take-home message of this set of data is that indeed GMC deficiency impairs retinal vascular perfusion during development, both at P5 and P14. Using electron microscope and immunofluorescence staining, we also confirmed this data. So, for example, here you can see some of the macrovascular in macrovascular vessels in GMC knockout mice. There is no blood. There is no lumen, or there is a very small lumen. So that confirms GMC knockout impairs the perfusion of the macrovascular in the retina or the lumen formation, right? So we also confirmed the inducible GMC mice had the similar phenotype. However, this phenotype was not dependent on the role of GMC as a junctional molecule, because endothelial junctions were normal and vascular permeability was not increased when GMC was deleted. So we thought this is not the reason. Then we did this RNA-seq again, and we found okay, when GMC is deleted or knocked out, and endothelial cell matrix relocation and the ECM organization was impaired. So then we confirmed that both in vitro and in vivo there was abnormal extracellular matrix declaration following GMC deficiency. Well, we also confirmed that GMC knockdown impaired endothelial cell matrix adhesiveness and suppressed the integrin beta 1 activation in endothelial cells. Well, downstream we found GMC, when GMC is present, it can recruit focal adhesion proteins, including NWASP and ARP3, and in turn activate integrin beta 1 signaling. That is why when GMC is deleted, focal adhesion formation is impaired. So I think this is the last slide, and we confirmed this set of data using integrin beta 1 EC conditional knockout mice, because we can again can phenocopy this vascular perfusion defects in integrin beta 1 EC knockout mice. Finally, we used a antibody that can specifically activate integrin. So we injected this antibody into GMC EC knockout mice, and we can rescue the microvascular perfusion defects found in GMC knockout mice to some extent. Also, this is the we think this can explain why GMC deficiency can impair the vascular perfusion function in GMC in integrin beta 1 in an integrin beta 1 dependent manner. So here is the final conclusion. We found GMC promotes retinal vascular formation and perfusion by regulating integrin beta 1 dependent endothelial cell matrix adhesion. The molecular mechanism involves NWASP ARP3 recruitment and downstream signaling pathway. Right. So with that, I would like to thank my team and ground support. Thank you so much.
Thanks so much. Any questions? I will start a quick one. Professor Zhang, is there any strategy to restore the endothelial matrix adhering or increasing integrin beta 1 expression in the future in clinical? Thank you.
To increase integrin beta 1 expression, well, I don't see a strategy that has been developed. However, as I mentioned in my work, we can use integrin beta 1 activating antibody. That is called TS216. That is a specific clone developed previously. Well, it can open the conformation of it can induce conformational change of integrin beta 1 that to the open conformation. So I think that antibody was developed to for clinical use, but it failed, I guess. But still, we in the study we can use that, right? So, but in the future, whether people can successfully use that in clinical, I mean, it depends, right?
Thank you so much. Any question?
Fuyang, can I ask a question?
Sure.
Hello,
Welcome.
hello, Professor Zhang. So I'm interested in your first part of the study, the SMAD4 in endothelial cells, and you studied pulmonary hypertension, right? So we had this endothelial inducible knockout of SMAD4 a long time ago, and I think what we observed is the adult angiogenesis is very much impaired in these knockout mice. So in the pulmonary hypertension model, do you think this angiogenesis is also relevant or not?
Well, I believe so. But you know, when we deleted SMAD4 between weeks 3 and 4. That is considered as adult deletion, right? So during this stage, lung angiogenesis has stopped, right? So That's why we in our mice we did not find like the density of pulmonary vessels are increased. No, we did not find that. However, if SMAD4 is deleted during a developmental stage, well, after birth, I think blood vessel density should be increased a lot.
Yeah, that's lethal. Yeah.
And that's lethal. By week 3, mice should die, because I think the cause of death is bleeding, I guess, right?
Yeah. Okay. Yeah. Thank you.
Okay, according to the schedule, Professor Zhang is the last speaker. May I ask Professor Yibing Wang summarize this section for us?
Actually, I will give that all responsibility to Junyi, please. I open it.
No, please, Professor Wang. So Dr. Wang is the vice chair of our council, the basic and the translational cardiovascular research of AHS. So let me, on behalf of Professor Zeng, to thank everyone to join us to support this session, and we discussed a lot, and 2 speakers discussed the advances in heart, and another 2 speakers discussed the endothelial vascular studies. Yeah, thank you very much. I think we had a very good session this morning, and Professor Wang, please summarize the session.
No, it's good. I'm just saying that, this is actually a very nice society activity for Asian Heart Society, and hopefully we'll continue to promote, especially the translational aspect in our field, and linking the basic science discovery with our clinical experts. And so, again, I congratulate to all the speakers, and hopefully we'll have another time to interact and to exchange our new progress. Thank you.
Sure.
Okay, thank you.
All right. Bye-bye.
Okay, bye.
Okay.
Okay, bye.
Bye.